part, I am very pleased to introduce to you Dr. Noel Mueller, who is an associate professor in the Department of Epidemiology at Johns Hopkins Bloomberg School of Public Health. And he's also a core faculty member in the Welch Center for Prevention, Epidemiology, and Clinical Research. Dr. Mueller completed his PhD in epidemiology at the University of Minnesota and completed his postdoctoral training at Columbia University. He conducts epidemiologic research at the intersection of nutrition, environmental health, and the microbiome, and aims to generate data to inform the development and testing of interventions to improve health throughout the lifespan. Dr. Mueller has been recognized for his research through the receipt of several national research awards, including the Sandra A. Daugherty Award for Excellence in Cardiovascular Disease Epidemiology, and was also recently recognized for his efforts in the realm of teaching and mentoring with the Advising, Mentoring, and Teaching Recognition Award from the Johns Hopkins Bloomberg School of Public Health. And so with that introduction, I'm happy to hand this over to Dr. Mueller. He'll be talking about the human microbiota, metabolites, and cardiometabolic diseases in pursuit of causality.
Thank you, Dr. Pern, so much for that introduction. I think I could probably skip some of the background I was giving you on my training and education because you gave such a nice introduction. So thank you so much. Wei, I've been, very impressed with your work over the years. We've overlapped with Project Viva, working with some investigators there. And also thank you to Dr. DeBalia and also Dr. Norris for the invitation to present to the Colorado School of Public Health, which has just been doing fantastic things and is under great leadership, clearly. So let's see, make sure my slides are advancing. Okay, so the outline for today's talk, I'm going to talk a little bit about myself, give you some background, although I want to make sure I get through all of my slides, so I'll probably keep that pretty short. I'll talk about some of my non-microbiome research because really microbiome research is one of the hats that I wear in kind of my research portfolio, but it's going to be the focus of today's talk. But I'm doing a lot of non-microbiome research as well. And then I'm going to go into kind of the focus of today's talk, human microbiota and cardiometabolic disease. Let's get the automatic slide advancing. That's unfortunate. And then I'm going to give a specific example about how the microbiome, unlike some other omics, lends itself to falsifiability or this potential outcomes framework of epidemiology. We can make microbiome observations from observational epidemiology studies that can be tested in experimental design, such as randomized controlled trials. So I'm going to tell you a little story about how we've done that with the human microbiota and child obesity. And then I'll talk about some other current and future research projects that we have going on, and then I'll leave you with a couple of concluding remarks. Okay. So can everybody hear me okay before I get going?
Good, yeah.
Okay, great. So a little bit about me. So I started my career in the Midwest. I'm from the Midwest originally. I'm actually from Wisconsin, but I started my career at the University of Minnesota, go Gophers. There I did a bachelor's of science in nutrition science. And then I took a year, actually I went to Colombia, South America, where I worked for a year in the United Nations to figure out whether I wanted to do basic science nutrition or public health nutrition or epidemiology. Came away from that experience knowing that I wanted to work in public health. So I went to pursue a master's in public health, thinking at the time I might want to do more public health practice, focusing with research in Latinos. I speak Spanish, so that was something that was of interest to me. But once I started doing my master's, I quickly realized that I was more of a researcher, and I worked with really good mentors at the University of Minnesota who kind of guided me through kind of epidemiologic research, but also some cluster randomized controlled trials in Latino adolescents. After my master's in public health, to just confirm that I wanted to do a PhD, I did 1 year at Georgetown University. So I did some work with Jeannie Mandelblatt at the Lombardi Cancer Research Coalition. That work was specifically focused on Latino populations and health disparities that they had around cancer and prevention and treatment of cancer. So we published a couple of papers, but I realized during that time that I wanted to be a PI of studies and not just do the research that other people were leading. So I went back to do my PhD. I chose to go back to the University of Minnesota because of the good mentorship over a couple of other programs, and did my PhD in life course epidemiology. During my time at Georgetown, I was also very well aware of the intractable challenge of getting people to change behaviors later in life after they already have chronic diseases. So I wanted to look at early life primordial prevention of disease. And I was very motivated by some of the work done by Dr. DeBalia in the Pima Indian study and life course approaches to cardiovascular diseases. After my PhD in epidemiology, I should say that my PhD dissertation was focused on the pubertal transition, so looking at the nutritional determinants of early puberty and then the cardiometabolic consequences of later puberty. After my PhD, I went to Columbia University to be a postdoctoral fellow on a T32 training grant that was focused on nutrition science and population health, kind of marrying the basic science of nutrition with population health. And I specifically wanted to move a little earlier in the life course to look at the early origins of cardiometabolic diseases and specifically focus on the microbiome, which was having a moment in science because of the high throughput sequencing technologies that had come out, largely had come out of Dr. Rob Knight's work at University of Colorado Boulder. So while at Columbia University, I worked with investigators in New York City. I found a natural mentorship with Dr. Maria Gloria Dominguez Bello and also Dr. Marty Blazer. They were doing much of the thought leading and pioneering work on early origins of the infant microbiome and how disruptions to the early infant microbiome may have long-lasting effects on health. I specifically was looking at how C-section delivery was impacting the microbiome and how those impacts may have long consequences for obesity risk. And I'll talk about some of that work in today's presentation. After doing my postdoc at Columbia University, I got a faculty position, a tenure track position at Johns Hopkins University, where I've been for about the last 7 years. I recently was promoted to associate professor. And at Johns Hopkins University, I've extended the microbiome research to look across kind of the life course, not just in early life, but also look at controlled trials in adults as well, looking at lifestyle interventions for weight loss, also metformin as an intervention, which happens to also affect the microbiome, and how the microbiome might be a mediator on disease outcome or pathways, but also a potential exposure. So we can take it and we can manipulate it and we can test whether there's a causal effect. Okay. So just some quick examples. We've really been a very, I think, successful group working together with postdocs, PhD students, and master's students to focus on cardiometabolic outcomes in pregnancy and also in early childhood. So this is just an example of some of the work that we've done looking at modifiable risk factors in cardiometabolic outcomes in pregnancy. Some of the major outcomes we've looked at are preeclampsia, but we've also looked at gestational weight gain and type 2 or gestational diabetes. And we've looked at, in terms of exposures, maternal diet, also exposure to heavy metals and trace elements and how the mixture of those can be teased apart and we can disentangle the effects of the individual metals. And then we've also looked at the metabolome more recently in the Boston birth cohort. And then we've also looked at cardiometabolic outcomes in childhood. This is just, again, an example of some of the research that we've done in this area, looking at, for example, how exposure to maternal plasma folate is a risk factor for elevated blood pressure later in childhood, being a protective factor. And also, for example, how exposure to ambient air pollution is associated with childhood blood pressure later in life. And the work that we've done has had an impact, and it has been covered by news media outlets and also has been cited by policy briefings, and also the World Health Organization has cited the work that we've done specifically on air pollution and child health. So I think that the work that we're doing is making an impact as well. Not only is it reaching academics, but it's also reaching people and making a real impact in their life. Okay, so now shifting gears, I'd like to talk about the focus of today's talk, the human microbiota and its role in cardiometabolic disease. So for those of you who are not well versed in the human microbiome or microbiota, the microbiota consists of the microbial communities, or is the microbial communities that live in our body and on our body, essentially living on all of our barrier surfaces or all of our epithelia. These include bacteria, fungi, archaea, and also viruses. While much of the work has been done on bacteria, there's also emerging areas or emerging lines of inquiry into the microbiome, which is the fungal microorganisms, and then also the virome. So we've learned a lot over the last 2 decades since the advent of high throughput sequencing. We've learned that the majority of the microorganisms that live in us and on us are actually not pathogenic, but rather commensal and symbiotic, potentially supporting our health. We've learned that microbial genes vastly outnumber our own human genes. We've learned that there's co-speciation of bacteria in particular with us as humans. That goes back as far as 15 million years ago, suggesting that this coevolution of bacteria with us as a species may be to help support our health. And we've also learned that unlike genetics, in which we share the majority of our genes with our neighbors, we actually have high inter-individual variability of our microbiome, suggesting that the microbes that live in and on our body may have a big role for the inter-individual variability that we observe with phenotypes and phenotypic differences between you and your neighbors. So we've learned that there's many essential functions for the microbiome, beginning with at the beginning of life, where the microbiome colonizes the newborn baby, educating their immune system. We also know that the microbiome is involved in regulating our mucosal barrier, not only our intestinal tract, but also all of our mucosal barriers. And it's directly involved in nutrition by extracting energy that wouldn't otherwise be used from indigestible polysaccharides, for example. It's involved in the production of metabolites and vitamins and minerals, which otherwise wouldn't be able to be produced if not for our microbes. What's particularly interesting about the microbiome for me as an epidemiologist is that I'm interested not only in understanding mechanisms, but also intervening on those potential mediators or exposures. And the microbiome lends itself well to this potential outcomes framework, where we can observe something with microbiome discovery, and then we can test or falsify our hypotheses about those different microorganisms. So the microbiome is largely determined by our environment. Only about 6%, it's estimated, is determined by genetics. And it's modified by lifestyle factors, environmental factors, pharmacologic agents, and it can also be modified, of course, by live biotherapeutics or probiotics. This slide is to just make sure that I mentioned that the microbiome is involved in a lot of these disease processes, and the major hypothesized mechanism through which the microbiome affects our health is through the production of metabolites. And these metabolites can be measured in the feces, but they can also be measured in the blood, where some are concluding that they may have even more direct effects on our physiology, causing disease states. And this is good for studies that haven't previously collected stool samples, but are still interested in gaining insight into potential microbially mediated pathways to disease. And some examples of microbial metabolites that you might be familiar with are short-chain fatty acids, but also bile acids. And P-cresol, for example, is a metabolite that's been recently discovered to potentially play a role in the development of autism. So there's a large or wider range of metabolites that are produced by microbes, and these can be picked up on by common platforms such as the platform from the Broad Institute, but also the platform from Metabolon, which now has a specific panel that looks at microbially produced metabolites in the blood. So now specifically the evidence for human microbiota and cardiometabolic disease, I think the strongest evidence that we have for the role of the human microbiome with disease outcomes as of yet comes from studies that have looked at the microbiome in an observational setting, found an association, and then actually taken the microbiome from humans, the human microbiota, and transplanted that into germ-free mice. So this experimental model, which was developed by Jeff Gordon and colleagues from Washington University, started with obesity back in 2006, but has extended to other outcomes as well, including insulin resistance, hypertension, atherosclerosis, and renal insufficiency, to mention a few cardiometabolic outcomes. And essentially, starting with the obesity model, what the investigators do is they look for monozygotic twins that are discordant on some disease phenotype. They'll take the microbiota from those discordant twins who share all of their genes essentially in common and transplant the microbiota into germ-free mice that can receive human microbiota. And what they find, for example, with obesity is that the germ-free mouse that gets the microbiota from the obese twin stays lean. This experimental model suggests that human microbiota are sufficient to cause obesity and these other cardiometabolic outcomes in mice. However, we still don't know whether this is true in humans, where the biological model of disease is much more complex, with not only biological factors interacting with the microbiome, but also socio-demographic factors, of course. So now I'm going to tell you a story about how we're starting to look or go from observational study to clinical trial to test whether the pioneering human microbiota, the very first colonizers, test whether they're causally associated with childhood obesity. So we know from work from 2012 was the first publication suggesting that the human microbiome develops in a dynamic fashion over the lifespan. But in the first 2 to 3 years of life, the human microbiome is in its most formative state and also most dynamic state. And once you hit about 3 years of life, it becomes more adult-like and stable, but can still change when you drastically change your diet or go on antibiotics. But during those first 2 to 3 years of life, when also the immune system is developing, the microbiome is in its highly formative or changeable state, okay? Research actually that came out of the University of Colorado from Dr. Stanislawski, who I think is on the call today, highlighted that differences in the early life microbiome over the first 2 years of life is associated with BMI Z-score at 12 years of life. And in this study of about 160 Finnish mothers or mother-child dyads, they found that the microbiome at, for example, 2 years of life explained approximately 50% of the variation in BMI Z-score at 12 years of life. And the variation explained was diminished, but still statistically significant at earlier time points, suggesting that the early life microbiome has an important role for the future development of BMI differences in childhood. Of course, this is one study, and future studies need to replicate this finding, but it does really open up the realm of possibility that the microbiome plays a large role in child BMI. It's also worth noting that in this study, higher alpha diversity at 10 days and 3 months early in the life course of the child and the development of the immune system, higher diversity was associated with higher BMI Z-score at 12 years of life. Now, this is different from adults, where higher diversity of the microbiome is associated with better health. In infants, there's more and more evidence suggesting that in the first 3 months of life, higher diversity is actually associated with poorer future health or poorer cardiometabolic health later in life. And I highlight this because I'll come to this later in our pilot study of vaginal seeding, where we're seeing this as an outcome. I just also want to mention, if anybody has any questions, feel free to interrupt me along the way. Otherwise, you can ask at the end. I'll hopefully save some time for that. So where does the microbiome come from? We wrote paper during my postdoc, which was describing essentially where we get our microbes from. Our microbes in a natural birth come from our mothers. The first exposure to the microbiome comes at birth. There was some evidence suggesting that there might be an in utero transfer of microbiota from mother to child. This was largely based on some evidence from 2014 from Kiersky Yagard and colleagues suggesting that there might be a placental microbiome. But since that study, several investigators have suggested that the placenta microbiome is largely due to either contamination or due to some other phagocytic remnants of bacterial DNA that are just existing in the placentas that they measured. So the largest kind of conclusion from that is that the first major exposure to microbiota comes at birth, and in the natural delivery process that comes either through exposure to ruptured membranes or through the long delivery process that happens with vaginal birth. And those first microbiota that are colonizing the baby, otherwise known as pioneering microbiota, are important because they are able to break down and biometabolize human milk oligosaccharides that are found in breast milk. Human milk oligosaccharides are prebiotics that are found in breast milk and otherwise not usable as energy by the infants if they didn't have the microbiota breaking them down. And some of the key microbiota that break down human milk oligosaccharides are Bifidobacteria infantis and also some strains of Bacteroides. So these early microbiota are thought to be important for essential nutritional breakdown of human milk oligosaccharides and may also have a role in metabolic health. So this vertical transmission of microbiota that happens is actually specific to several different types of bacteria. This is a study from Corpella and colleagues, which showed that the vertical transmission of bacteria was specific to strains that are in the family of Actinobacteria and Bacteroidea. And they showed that there was much higher mother to newborn sharing or mother-child sharing of these families of bacteria compared to Clostridia over the first 2 to 10 years of life, and then after that, there was some convergence. So this is consistent with this notion that mother-newborn sharing of Bifidobacteria and Bacteroides species is important for that early life nutrition that I was talking about. So what happens when we disrupt this mother-to-newborn microbial transmission? So as a postdoc, we examined the first stools, meconium, and then also transitional stool of C-section delivered babies and vaginally delivered babies that were born in Brazil. And we found, consistent with some other studies at the time, that C-section delivered babies had a very different microbiome than vaginally delivered babies. Here in this principal coordinate analysis plot, you can see that vaginally delivered babies cluster in this blue cloud, and C-section delivered babies cluster in this pink cloud. And these two groups are statistically different in terms of their phylogenetic composition of bacteria overall. When you start to look at what specific types of bacteria are different between the groups, you find that in general, and there's been over 25 studies now that have shown these differences between vaginal and C-section delivered babies. In general, C-section delivered babies are colonized by more pathobiontic bacteria, including Clostridium perfringens, Klebsiella species, and C-section delivered babies, relative to vaginally delivered babies, have fewer Bifidobacterium species, Bacteroides species, and at the very beginning of life, fewer Lactobacillus species. And we also showed that in this paper published in the British Journal of Obstetrics and Gynecology in 2020, we showed the same pattern was true in infants from the nurture cohort, a cohort of low socioeconomic status, low income, and racially diverse cohort. And this was statistically significant and quite strong. And this is at 3 months of life. In addition to differences in the microbiome, in the nurture cohort, and also in the New Hampshire birth cohort, it's been found that C-section delivery impacts the microbial metabolites And specifically, one microbial metabolite that's impacted by C-section is butyrate, with differences at 3 months. Higher butyrate actually was observed at 3 months in C-section delivered babies compared to vaginally delivered babies. And this was also found in the New Hampshire birth cohort, but there's also several other microbial metabolites that were impacted by C-section delivery in the New Hampshire birth cohort. So this suggests that there's potential biologic plausibility to not only there being differences in the microbiome, but these changes in the microbiome being related to metabolic outcomes. So far, we've really only looked or presented only data looking at how C-section or interruption of the vertical transmission is associated with differences in the microbiome and some microbial metabolites. But is this a public health concern? Ultimately, we're interested in whether it's associated with disease phenotypes. So overall, globally, there's been an increase in C-section deliveries. There's a recent commission by the Lancet to do an overall review of the literature, and they found that C-section delivery increased from about 12.5% in 2000 to over 20% in 2015. The overall averages were brought down by some low and middle income countries that still had very low rates of C-section. But in the US, the C-section rate is currently around 33% and has increased about 80% from 1990 to 2010. And in some parts of the world, such as Brazil and China, C-section rates are over 50%. So even if C-section is associated with disease outcomes with a relatively low relative risk, on a global or population-based level, that association can be quite meaningful for population or attributable risk due to C-section, even if it's a small relative risk of association with disease outcome. So in terms of disease outcomes that have been associated with C-section delivery, we documented that C-section delivery is associated with higher risk of obesity in childhood at age 7. So we found a 46% increased risk of childhood obesity in the mothers and children study of Northern Manhattan and South Bronx. This is some work that I did when I was a postdoc at Columbia University. And this is after adjusting for potential confounding variables and after adjusting for breastfeeding duration as well. We also, this is some work that I did at Hopkins, found that C-section delivered babies over the first year of life have a greater gain in weight for length Z-score, suggesting that they, even though they start at the same place, they gain more weight in that first year of life. And this may be on the pathway to them having greater risk for obesity later in life. In a cohort study from India, we had data on the type of C-section delivery, and we found that elective C-section deliveries had greater BMI Z-score, and also this persisted before and after adjustment for potential confounding variables, suggesting that elective C-sections, the association may be stronger with BMI Z compared to emergency C-sections. Many emergency C-sections are exposed to ruptured membranes or some of the labor process. So this is consistent with the hypothesis that it may be due to microbial sharing at the beginning of life. You still might be wondering whether this is a potentially causal association or due to some type of indication and the reason by which cesarean delivery may be called for in the first place. So it does take alternative approaches to look at this association. Jorge Chavarro and colleagues from Harvard were funded to get an NIH R01 grant to look at the association between C-section delivery and obesity and other cardiometabolic outcomes in the children from the Nurses' Health Study. And they used an interesting approach. They took a sibling design and looked at within the same mother whether a child born to cesarean delivery versus vaginal delivery had a different risk of obesity in childhood, adolescents, and adults. And what they found was that, first of all, cesarean delivered children compared to their vaginally delivered siblings had a 64% higher odds of developing obesity in childhood or adulthood. And then when they flipped the question, they looked at just vaginal births after C-section. They found that children born vaginally after C-section had a 31% lower risk of developing obesity in childhood or in adulthood, suggesting using a different approach that this association may be causal, okay? But ultimately, we can't get at causality without experimentation. There have been some experiments in mice. This is a study done by Maria Gloria Dominguez-Bello and her postdoc Keith Martinez, looking at whether C-sections associated with change in body mass index in mice. And what they found was that C-section delivered mice had greater accrual of body mass over the first 15 weeks of life compared to vaginally delivered baby or vaginally delivered mice. And this was consistent in females and males, okay? But again, this is just in mice. So how can we, okay, and before I get to the next part, I also just want to mention that C-sections also have been associated with several other health outcomes in addition to obesity. I'm just focusing on obesity here. That's been the majority of the research that we've done. But if anybody's interested, there's some literature looking at C-section delivery with autoimmune diseases, food allergies, and also ADHD and autism. And this is, I'm only citing here systematic reviews and meta-analyses or large Scandinavian cohort studies of over 1 million mother-child pairs. So back to the story. So experimentation in mice is one way to look at causality in mice, but if we want to look at this in humans, how do we test whether or not the pioneering microbiota and disruption of vertical transmission is potentially causal? Well, we can take one aspect of delivery mode or of this process, which is the microbial sharing, and we can test whether microbial sharing through microbiome transplant is associated with changes in the microbiome and eventually changes in disease risk. So there was a group who did this in a non-randomized setting in Finnish mothers and children. They actually looked at whether maternal feces could be used to restore the microbiome of C-section delivered infants compared to vaginally delivered infants. So what they did was they provided the C-section delivered infants with fecal microbiota from the mothers, from their same mothers, and compared them to either C-section delivered infants or vaginally delivered infants. And what they found was that those infants that received the fecal microbiome transplant from their mother actually did have a microbiome development that was more similar to vaginally delivered infants in that study compared to C-section delivered infants. And they looked at Bacteroides, Bifidobacterium, Clostridium, and also several other species, and found more or less the consistent message that there was some restoration that could happen if you were to take the intestinal microbiota from the mother and transplant it to a C-section delivered baby. However, the pathway for moving this or generalizing this in the US, I think, is very murky, especially given that there's been even a halt on fecal microbiome transplant studies in adults because of the risk to adults for infections, and also that fecal microbiome transplants potentially led to several patients' deaths. Whether or not those were causal or not is still being debated, but there was a halt. And I think putting fecal microbiota into otherwise healthy newborns is going to be very, very difficult. And whether or not we even want to do that in the first place is another question. Another approach, which potentially is more benign, is sharing vaginal microbiota with the infant. And this is an approach known as vaginal seeding. And it's depicted, kind of how it's done is depicted in this cartoon. So before antibiotics are given for prophylactic C-section, prevention of infections from C-section, before antibiotics are given, we're doing a study of vaginal seeding. Now, what we do is we give mothers a gauze in which they inoculate the gauze with the vaginal fluids before antibiotics are given. The gauze is then kept in a sterile container. And then at the time of birth, the clinician or the nurse or the practitioner wipes the baby's mouth, wipes the baby's face, and then wipes the rest of the baby's body before returning the baby to the mother, where they have skin-to-skin contact if that's possible. So this is just a process of vaginal seeding. In a non-randomized trial of vaginal seeding, Maria Gloria Dominguez-Bello, who really kind of pushed forward this whole notion of vaginal seeding, She looked at whether vaginally seeded babies compared to vaginal or C-section delivered babies had differences in the microbiome. And what she found was there was partial restoration, in her words, of the babies that were inoculated with the vaginal microbiota. So they started to resemble those vaginally delivered babies more so than C-section delivered babies. And this restoration occurred for several species that were in general missing from C-section delivered babies. So Bacteroides was partially restored, Lactobacillus was partially restored, and then also in addition to being restoration in the infant gut, there was restoration on the infant's skin as well. But I'm highlighting that this is a non-randomized trial because women who elect to do the vaginal seeding may be fundamentally different than women who have C-section and don't want to have vaginal seeding done, or the vaginally delivered births. Again, this is more or less an observational study suggesting that there could be changes to the microbiome. Because of these observational studies, the American College of Obstetrics and Gynecology had to make a statement about whether or not they approve vaginal seeding for women who are delivering by C-section. And we are in agreement with their recommendation, which is that we need more randomized controlled trials to prove safety and the potential health benefits of vaginal seeding, and whether potential health benefits outweigh any potential risk. All we know at this point is that vaginal seeding may restore some of the commensal and symbiotic microbiota. However, we have very small sample sizes, and it's only been observational studies. And this is important because women already are requesting vaginal seeding, and also women are already performing vaginal seeding at home. If we don't do anything, don't have any clinical trials, then this will continue, and there's potential for this to be something that causes infections at home, okay? So we've developed a randomized controlled trial rigorously designed to have a double-blinded randomized controlled trial so that mothers and the clinicians are not aware of the randomization assignment. For the trial, we enroll elective C-section deliveries and randomize them to either the babies to receive maternal microbiota through the gauze or to receive a gauze that's sterile. And then after that, follow the babies to see whether there's differences in development of the microbiome, and then also in the long run, differences in phenotypes that have been associated with C-section delivery. So far, we have interim data suggesting that there are differences in the microbiome at both day 2-3 and day 30. In a seeded group, we find that the group that's seeded with the maternal microbiota have lower Shannon diversity, a measure of alpha diversity, at day 2-3 and also at day 30, which again is consistent with this notion that those babies that are colonized by bifidobacterium or Bacteroides tend to have a predominance of these bacteria, and the predominance of this bacteria can keep down the overall diversity of the microbiome, and this has been suggested to be beneficial for health later in life, and I'm happy to go into that more in that Q&A discussion. What we also found was that when we sequenced the gauzes from the mothers, we found that the vaginal microbiome in pregnancy is not just vaginal microbes, but also contains microbes that are typically found in the gut. About 58% of the microbes that were in the vaginal sample were also found in the maternal gut, suggesting that during pregnancy, unlike outside of pregnancy, the vaginal microbiome starts to resemble more the gut microbiome. And this has been suggested by other studies also, including a recent publication from Song and colleagues, in which they suggested that the vaginal microbiome during pregnancy, particularly around the time of childbirth, starts to resemble more the fecal microbiota, and this may be an evolutionarily adaptive change to the vaginal microbiome so that they can share with their child those microbes that are important for the very beginning of life. Because we're interested in whether the kind of microbiome of infants are colonized by maternal vaginal microbiota or fecal microbiota, we applied for an Opportunities and Infrastructure Fund grant from the ECHO Consortium. and were awarded this. And now we have funding to look at the ECHO-wide platform for studying mother-to-infant vertical transmission of microbiota in the ECHO study. The ECHO study, for anyone who's not aware, is a large study that comes directly from the NIH Director's Office. It brings together cohort studies throughout the US, approximately over 70 studies from throughout the US, representing a wide array of sociodemographic groups, racial and ethnic diversity, and also different life stages. And so many of the cohorts have been able to measure the microbiome of mothers and also infants during the first year of life. So the objective of this ECHO-wide study is to identify the maternal microbiome site that is most responsible for the vertical transmission of microbes from mothers to the infant gut. Our hypotheses are that mother-child pairs have higher concordance of microbiota than unrelated pairs. We also hypothesize that maternal gut microbiota are transmitted more to a greater extent than maternal vaginal microbiota, and that maternal prenatal antibiotics and C-section delivery modify the sharing of bacteria from mother to infant. And so far, we've had positive responses from four cohorts in ECHO that have identified that they have the data from mothers and infants to contribute to the ECHO-wide analysis. Okay, so I think I got about 5 more minutes, so I'm going to go quickly through the rest of my talk. Other current research that I just wanted to note, I'm going to end kind of my story about the early infant microbiome and childhood obesity, switching gears to other research. So this is my KO1 research, which I have funding for about the next 2 years, taking into account a 1-year no-cost extension. And my KO1 research, different from the early life microbiome, is getting at whether or not the associations from observational studies around diet and the microbiome and short-chain fatty acids are potentially causal. So we published several studies from using the funds from the KO1. We looked at, because there's stored samples from several DASH studies. DASH is a dietary approaches to stop hypertension. We've been able to look at stored samples and the effect of different types of DASH diet on short-chain fatty acids, the microbial metabolites, and publish this research. One key finding is that when you switch from a typical US diet to any version of the DASH diet, whether it be high carbohydrate, high protein, or high unsaturated fat, there's increases in acetic acid, one key microbial metabolite. There's also increases in butyric acid, but these are greatest for protein. And then there's decreases in some of the other short-chain fatty acids. So there's clear changes that are happening when you switch from a US diet to a high fiber DASH diet, whether that be high carbohydrate, high protein, or unsaturated fat. But that was a controlled feeding study. When we then ran a new randomized controlled trial looking at a coach-directed weight loss arm that was coached to be on a DASH diet compared to a self-directed arm. And then another arm of the study was metformin. We found that the coach-directed arm in that study design did not have strong effects on the microbiome. And we attribute this to the coach-directed weight loss arm not having a controlled feeding component. It was just suggested that they take in the DASH diet. However, what we did find in this study we were able to publish on is that metformin had a strong effect on the microbiome composition, both at 6 months follow-up and also at 1-year follow-up. And I'm not going to get into the details of that, but that's published in Diabetes Care, and I think that's another interesting line of investigation. What we did find, interestingly, is that the coach-directed weight loss arm did have effects on short-chain fatty acids, the same short-chain fatty acids we measured in the OmniHeart study that I presented before. And that we also found that changes in short-chain fatty acids, both in the feces and also in serum, are associated with changes in blood pressure and also prevalence of hypertension in the SPIRIT trial. One of the consistent findings was that increases in butyrate over the course of the trial were associated with decreases in blood pressure. And this is a paper that's coming out in a couple of days in the Journal of American the JAHA, or Journal of the American Heart Association. Another finding I'm just going to mention here is that we found that the coach-directed weight loss arm had reductions in a measure of gut permeability, lipopolysaccharide binding protein. However, there are no changes in this outcome measure for the self-directed or the metformin arm. Okay, that was just a quick overview of some of the work that's come out of the KO1 award. I just want to quickly touch about what we're going to do to now build off of the KO1 award. One really exciting new development is that we're getting funding to do development of a new smart sensor for microbial gas production. And then also we get to test that in a randomized controlled feeding trial. So this is a mechanism known as the R21, R33 mechanism. 2 years of the R21 are for development and preparation for the R33 phase. The R33 phase is equivalent to R01 type funding, and we're doing a controlled feeding trial. And this will be starting shortly. Another grant that I've submitted is a grant to look at the effect of dietary patterns and sodium intake on the gut microbiome and the metabolome of adults with type 2 diabetes. This is from the DASH4D parent trial, which is ongoing, and we're collecting stool and blood specimens so that we can look at the microbiome and the metabolome in these adults with type 2 diabetes. There's still very little evidence about how different dietary patterns and also how different levels of sodium intake impact the microbiome and the metabolome. And then a grant that we're preparing for submission in July, which I'll be the primary PI for, is the association of the gut microbiome, microbial metabolites, and Alzheimer's disease in the Tobago Health Study. And then we'll also be using the Baltimore longitudinal study of aging to validate the findings from this study in Tobago. Okay, and then I just wanted to brainstorm potential collaborations with faculty at Colorado School of Public Health. So a couple of what could be potential low-hanging fruit, I think, would be collaborations on the MAX-Y study. I know Dr. Abraham works in the MAX-Y study, and Dr. Erlandson, who's worked with one of my colleagues, Todd Brown, has also worked in the MAX-Y study. And also the ALIVE cohort. Both of these cohorts I identified because they have microbiome samples available, and they also have some measures of neurocognitive function and could potentially add other measures. I think that this is an interesting area, and there's a recent RFA supporting that this is an interesting area because populations with HIV have much higher risk of Alzheimer's disease and other neurocognitive outcomes. So this might be an interesting potential collaboration. And then another potential collaboration with faculty from the Lead Center, including Dr. DeBalia and Dr. Wei Peng, would be looking at fecal microplastics, microbiota, and microbial metabolites and their role in fatty liver disease in adolescents, potentially using samples from the EPIC cohort. But I think this is just kind of scratching the surface. I see a lot of really nice kind of synergy between the work that I'm doing and the work that people, faculty at the Colorado School of Public Health are doing. And I would be very interested in talking more about potential collaborations, moving forward. So I'll just make a couple concluding remarks. The human microbiota, as hopefully I've been able to convince you in this talk, are highly modifiable and associated with cardiometabolic disease, potentially in a causal way. Some of the major modifiers I've shown here, and we've published on many of those. I also think microbiota are unique from other omics because observational findings can lend themselves well to falsifiable hypotheses with experimental designs. I've shown you in this presentation one kind of way that we've gone about doing that using the early life microbiota with childhood obesity. But in the future, discoveries, for example, between the microbiome and Alzheimer's disease can be tested by collaborating with people who do live biotherapeutics, and we can randomize a consortia of microbiota to see whether it alters metabolite pathways or even disease phenotypes such as Alzheimer's disease later in life. So future interventions specifically include microbiota-focused diets, next generation live biotherapeutics, prebiotics, postbiotics, and even microbiome transplants, but these need to be rigorously tested with randomized controlled trials. And then I also just want to point out that the microbiota may be leveraged for disrupting things such as the intergenerational cycle of obesity and cardiometabolic diseases, and even for interventions to remediate environmental exposures, such as kind of exposures that are very difficult to remediate otherwise, like lead or air pollution. Could be opportunities for collaboration there as well. And I also finally just want to leave on this note that there's a lot going on in the microbiome world, and a lot of that work is being done by bench scientists. And I think the role for epidemiology, getting involved in microbiome science is huge. It can help with study design, and there's a lot of opportunity for discovery still. So I hope that you agree with me and consider doing more microbiome epidemiology in the future. All right. Before I end, I just want to say thank you to many of the mentors and people who are on the Mueller lab, including Mingyu Zhang, Maura Differding, Tiange Liu, Sarah Benjamin Nealon, Chaobin Wang, and Lawrence Apple have been big mentors to me. And then also, of course, the grant support that I've received along the way, including a career development grant from NHLBI, a Mid-Atlantic NORC grant that helped to fund a lot of the research on the early infant microbiome development, and then the Opportunities and Infrastructure Fund from ECHO. And then finally, thanks to all the participants, of course, without whom none of this research would have been possible. And thank you to everybody else for your attention. I'll take any questions. I couldn't end this presentation without showing some pictures of my kids, who are really the light of my life. So thank you so much.